Okay. Hi, good morning to everyone. It's nice to see everyone bright and early this morning. Uh, I'm joining you from Taiwan today, actually. I'm in Taiwan for the rest of the week just for work purposes. But through the miracle of technology, I can still join you regardless. So it's very nice to see everyone bright and early. Nice to see our usual faces and a few faces that I don't get to see very often. Chingling, hi, good morning. I didn't get to greet you during the ECCP luncheon with Yusek Merly Sombrilla, but Mercy
Oh
Sombrilla, but
yeah.
you left before I could say hello.
Okay. Well, will I
Oh
trump you?
well.
I'm joining from Toronto.
Okay, so at least I'm in the same time zone. You're
I
not.
know.
Okay.
Okay.
Okay, anyway, so good morning to everyone. It's nice to see everyone here bright and early, or in Chingling's case, heading into the night. I am
Yep.
your chairman, Julian Barcelona, and I'm very glad to join you for our last session for July. Just a quick reminder to everyone, after this session, we're going to have our General Assembly. So I encourage everyone to please, all of our members to please stay behind for the General Assembly after this session. The General Assembly will take the place of our informal session after this. So while normally we would have an informal session from 9 to 10. This time it will be replaced by our General Assembly. So we can go a little over time with questions if in case they're quite tough. Okay. Anyway, as always, we start our sessions first by thanking God for bringing us together. May we please start our session with a short prayer.
Lord, thank you for this day, for the presence of everyone in this call. For once again, we are gathered and given the opportunity to appreciate life and your words. Forgive us for our shortcomings and cleanse our hearts as we learn and live your words and example. Father, bless our endeavors, especially today's meeting. May you guide us in our discussions, and may you enlighten our minds in every decision that we make. Give us your grace that we may effectively do our parts for your greater glory. All this we ask in Jesus' name. Amen.
All right. And after God comes country, may we please give our respect to the Philippine national anthem.
Mga kababayan, ang pambansang awit ng Pilipinas. Kayang magiliw, helas ng sinananan, alam ng puso, sa titik mo'y buhay. Lupang hinirang, tuyag ka ng magiting, sa manluluping, di ka pasisingil, sa nagagang tutok sa simoy at sa langit ng pangaw, ay hinagang tulakaw, sa pagkayang minamahal, ang isang ng matawad mo'y tagumpay na naghihindig, ang bituin at araw niya kailan pa may magtitindig. Luman ang araw ng walang pagsinta, buhay langit sa piling mo. Ang hindi kaya ng pagkay mga amin, ang mamatay ng dahil sa'yo.
All right. And after country comes the people that make everything happen. May we please flash on screen the generous members of the Agribusiness and Countryside Development Foundation. We are a member-supported nonprofit organization, and without the generous support of all of our members, our special partner institutions, our senior special partner institutions, corporate members, and individual members, we would not be here today to bring you our sessions every week. So we thank each and every single one of you for your generosity to allow us to continue working for agribusiness and countryside development in the Philippines. And for those of you who are joining us this morning, once again, good morning. I am Julius Barcelona. I am your chairman. It's a special session today because after this session, we're going to have our General Assembly. If you're joining us 2 weeks later through our social media uploads, welcome. We hope that you enjoyed today's session. As I mentioned, we are a member-supported nonprofit organization. And if you like what we do and you'd like to support us, as well as gain access to our wide network of countryside agribusiness and countryside development organizations, consider joining us as a member. We'd be very happy to welcome you into our fold and welcome your support greatly for our activities. Our links are in the description. You can send us an email to introduce yourself there. So without further ado, may I please ask our president, Mr. Ernie Ordonez, to introduce our speaker first.
Yes, good morning to all of you. We'd like to say that in recognizing all our partners, I want to give special mention to Multinational, who's given us the gift of Bernie Up, who for 14 years has done something nobody else has done in the country or in history, which is every week forum. Multinational and Bernie Up, of course. Today, my task is to introduce to you our guest speaker, Dr. Nonawin Lucob-Agustin. I'd like to say this is very appropriate because yesterday in the SONA, President Marcos talked about the danger of climate change and water. And today, our topic is precisely that, what will happen to our crops with different degrees of water coming to us, especially with El Nino coming. So our topic is measured from the underground. In reality, it talks about water stress, and of course, that's underground and how our crops will come to it. But more interesting for me than the topic is the speaker, right? Speaker is the youth personified, the new youth. And I like to say that I want to compliment again this board, which before was over 60 years old. Now everyone is below 45, except one. And basically, she epitomizes the new spirit of the board. And this new board, actually, which will be doing a review in a few minutes, has been responsible for the participants, participant hits participation, going from 540 a month to more than 8,000, right? It goes from just Manila now to the whole world. 20% of whom are abroad, like participating here. We have our chair in Taiwan right now. We have Tingling, Tanko, who's in Canada. It's really worldwide, right? And of course, as our chairman said, our job is not just to transmit technology, but getting the action from that. And we're asking Dr. Nona during her speech, to give us 5 minutes of her personal, like how did you get there, right? Personal interaction, because we intend to work with her between forums as we work with all the speakers to fulfill the objective of our department chair, which is do action from the forum. And of course, 5 minutes personal life, and then her phone number so we can contact her from wherever in the world. And of course, how we can help her with her mission, because we want to act on what we learned today, right? Having said that, who is Dr. Nona? Well, Dr. Nona is youth personified. She's a body scientist, not like other people who go abroad and disappear. She's come back. More importantly, she did her agriculture in UP Los Baños, of course, fantastic school. But her master's and doctor's degrees are from Nagoya University in Japan, where she came out as an outstanding person. And she put that into practice. She has publications here as co-author here and abroad. And of course, being a big scientist, she is sponsored by Science and Technology. But in addition to all these things, she was recognized in Asia as 5-year women leader program in Asia. And so she's global. And that's what we have to say, we have global. So even in our agriculture, we have to global. With this challenging background, we'd like to welcome Nona. Nona, please start with 5 minutes how you got into this, so we get a personal touch, and give us your phone number so we can call you from all over the world. And in addition, towards the end or in between, tell us how we can help you in your mission. What we have here is a group of people who are business-oriented with contacts with government and banks and other businessmen and farmers. And we are, of course, now with the legislative body by Congress called the Field Agriculture and Fisheries Council, and we've been elected the chair of the International Trade Committee. So we bring the business aspect into this, and they say we need that very much. So we need science and business. We need science because if we don't have it, like we don't have to do with water, which you teach us, we can really not progress. And we need business because unless we have business, we will not be sustainable. So you have this thing all wrapped up. So without much further ado, we'd like to have you all listen to Dr. Nonawin Lucob-Agustin, who is like an angel from above, coming from abroad, winning all the awards, studying in Japan, etc., and back with us in our country. Nona, the floor is now yours.
Yes, good morning po sa ating lahat. Thank you so much, sir, for that very generous and kind introduction of me. So I actually started research at the Philippine Vice Research Institute, working on particularly root research. And after that, after 2 years, I went to Japan for further graduate studies. And I also continued my root research there, focusing on more of genetics, very basic studies, genetics, epigenetics, and plant breeding. And right now, I am given the opportunity to come back to Philippines and do breeding and biotech applications of my research works. And I hope that I will be able to share all those information and the things that we are doing in order for us to also contribute to our SNT, the Science and Technology and Agriculture in Philippines. So, shall I share my screen po, sir?
Go ahead.
So that I can share my research. Yes. So I prepared my PowerPoint presentation, which highlights the researches that we have been doing and we are advancing. And I hope that by sharing this, we can also be able to do some collaborative works on how to further advance and do maybe business related collaborations in this research. So is it already okay? Is my presentation okay na po?
We can.
Yes.
So once again, good morning po. It's a pleasure for me to present to you our recent works on this particular topic, and hopefully we can be able to give new insights on the importance of root research and its usefulness for breeding and biotech applications. And we are looking forward actually for the possibility of new collaborations with your group. So this research is a collaborative work with my co-authors from Phil Rice and Nagoya University, Japan. So let me start off by bringing your attention to this figure. In here, we can see that rice is cultivated in different environments according to the availability of water. And this is very timely to the message that Sir Ernie have said a while ago. So we have the upland rice at the highest topography, the irrigated rice, which we usually see for our rice fields. We have the rain-fed lowland rice that looks like this irrigated lowland, but the source of irrigation is purely coming from rainfall, unlike this irrigated lowland whose source of irrigation is from the irrigation canals. So this rain-fed lowland has a problem. Then we have the flooded environment for rice at the lowest topography. And among these rice environments, the most productive ecosystem is the irrigated lowland. But we have also many non-irrigated areas that are still planted with rice which we would like to address in order to contribute for production, especially in our country. So we can also observe here that rice has evolved its root morphological features in order to adapt to the prevailing environment in each ecosystem. I hope you can see my cursor po, no? So in Philippines, yes. So in Philippines, we have two important production of rice, the irrigated and the rain-fed lowland. And as I've mentioned, this rain-fed lowland is an unfavorable environment due to its reliance to rainfall as source of irrigation. And it occupies 31% of the total rice production devoted, of the total area of rice, of the total area devoted to rice, and these are actually distributed across the country. So we expect that the annual production is also lower compared to the irrigated areas, and that is 4.7 million tons in rain-fed, while 14.6 million tons in irrigated. So similarly, the irrigated ecosystems are now threatened by drought due to climatic changes. And as shown in this map, almost all areas in Philippines are vulnerable to drought. In here, the more intense the color indicates, the higher the drought vulnerability of the area. So in fact, in 2016, we have 85 provinces affected by El Nino, and some of these provinces even declared a state of calamity. And in 2019, El Nino caused 5 billion production loss in rice and corn. So this is very interesting because it's also timely to the message of our president. And I hope that our research are really contributing in addressing the concern on El Nino. So as I've mentioned, irrigated rice fields are also vulnerable to drought, probably because the water levels of our dams have already decreased. And actually, those rice fields at the tail end of the irrigated system are already exposed to drought. So for instance, these rice fields are cultivated as irrigated areas for 20 years, but they are now experiencing water scarcity. And we can see that drought has affected the growth of the rice. So they are very short, and of course the yield are reduced. And in worst case scenarios, the farmer decides not to plant rice anymore if there are no water available. So considering those scenarios, a possible solution for rice to adapt is roots. And why roots? Because roots are really very important to plants, especially when we are discussing about the availability of water and nutrients which are in the soil. So in the 1960s, we have the so-called Green Revolution, which simply focused on the shoot and yield. Whereas because these roots are hidden underground, they are considered the neglected other half of the plant architecture. But recently, roots gained an attention to many researchers, especially the plant breeders, because it is really one of the ways to improve or support the above ground traits to sustain production. So regardless of favorable or unfavorable environments, roots are very important, and desirable root traits are needed in order to function properly and support the shoot or the above ground parts of the rice plant. So we particularly focus on the highly developed branch root system or root plastic development as an adaptive trait of rice for drought stress. So this is important for the expansion of the root system for water and nutrient uptake, and thereby the maintenance of stomatal conductance that contributes significantly to the maintenance of dry matter production, whether shoot or yield, under drought stress condition. So the promoted roots or improved root system through high branching maintained this shoot and yield by maintaining the actually the water and nutrient uptake, no? So before I go further, let me just show a photo of this rice root system and its different components. So the root system of rice consists of mainly the crown roots, which is the crown roots and lateral roots. And the seminal root is actually the first root that comes out from the plant, followed by crown roots, which are produced from the stem. And from these seminal and crown roots, the lateral roots are produced. And these lateral roots are further classified into distinct types. We have the S-type and the L-type lateral roots. So the S-type lateral roots are short, thin in diameter, and they have no capability of higher order of branching. Whereas these L-type lateral roots are thick, long in diameter, and they have the ability of doing or producing higher order of branching. So the L-type, these two lateral root types are actually they have actually big difference in terms of their development and function. So the L-type lateral roots are really the important component of root, especially during drought, because these roots are efficient in taking up water from the soil. So regardless of upland or lowland, the lateral root branching ability is important for enhanced water uptake. So what do we need? We need to breed high-yielding rice with improved root system through promoted L-type lateral roots. And in order to fast-track breeding, the selection of lines and genetic improvement, we need to identify the genes and molecular mechanisms associated with promoted L-type lateral root formation. So there have been studies about the genetics of root system formation through the availability of mutants and the isolation of their respective positive genes. However, most of these mutants have defects on any of the root components. So the utilization of the L-type lateral roots as index for breeding has been limited, and these mutants cannot be used for breeding. So what we did now was to isolate mutants with a promoted root system. And these are the WEG1, the PLR1, and R1 mutants. So the WEG1 mutant had a wavy parental root that produced high number of L-type lateral roots as compared to this wild type. And on the other hand, the PLR1 and R1 mutants had highly developed lateral roots or developed a root system as compared to their wild types, Nippon Bare and Kimase respectively. So I will explain for the mechanism of these mutants one by one. So in WEG1 mutant, we observe clearly that the L-type lateral roots are produced from this outer side of the bent region. So to determine the causative gene of the wavy root phenotype, we checked the cell division, and we found that the cell division at the root tip of the wild type and WEG1 were the same. So this means that the wavy root phenotype of the mutant is not determined by cell division. And next, we check the elongation zone, the elongation rate at the elongation zone. And we can see that the cortical cell layers at both sides of the mutant were the same, whereas the cortical cell lengths at the outer side of the mutant were longer than the inner side. So these results suggest that the WEG1 root phenotype of the mutant is determined by this asymmetric cell elongation, but not cell division. And so we further isolated the causative gene of WEG1 on chromosome 9 by map-based cloning, and the causative gene was located on these two markers wherein a single base nucleotide substitution difference was revealed. So this mutation is actually at the fourth intron or non-coding region that resulted to a protein change, and so we can observe this change in the root phenotype of this mutant. So the gene is annotated as a member of the HRGP superfamily, or known as cell wall proteins, and there is no report on this gene on the regulation of elongation, but some of the members of the HRGP were reported to control cell elongation. So we performed QRT-PCR of root samples from different root zones, and we have shown here that this gene is highly expressed at the elongation zone. And the WEG1 expression pattern is actually coinciding to the expression pattern of GUS, and this WEG1 is localized at the elongation zone and expressed at the cortical cells. So this means that HRGP is important in the normal regulation of cell elongation. Then we further observe that the root hyoxine accumulated at the outer side of the bent region of this mutant, as shown by these green dots, which suggests that a high amount of auxin is needed to induce the L-type lateral root formation. So these are our results, and I would like to recap. At this point, the WEG1 mutant showed wavy root phenotype, and the causative gene is an HRGP member known as cell wall proteins. It is expressed at the cell elongation zone at the cortical cells. So this mutation disrupted the elongation zone wherein the cortical cells at the outer side of the bent region were longer than the cortical cell lengths at the inner side. So that's why we observe this wavy root phenotype of WEG1, and auxin accumulation induced the L-type lateral root formation. So next, we found the PLR1 mutant with developed lateral roots referred to as PLR1 mutant, or promoted lateral root. So to understand the mechanisms, we also isolated the causative gene of this mutant on chromosome 9, and we revealed that the causative gene is a single nucleotide substitution from G2A, which resulted to the amino acid change from glycine to glutamic acid. And the causative gene is the OSMED25, which encodes a member of the mediator complex, which is known to interact with transcription factor and recruit the RNA polymerase II in the regulation of the target genes for transcription. So as I mentioned, the mutation gene of PLR1 mutant was induced by one amino acid substitution that is found on the acid protein domain, which regulates transcription factor. So through this domain, the OSMED25 interacts with transcription factor. So this means that the mutant cannot interact with the target transcription factor. So in the case of the wild type, the mediator complex interacts with the transcription factor and recruits the RNA polymerase II, and finally the transcription occurs. And we thought that this target gene negatively regulates L-type lateral roots, because in the case of mutant, they produce high L-type lateral root number, and the mutation of this gene did not interact with the transcription factor. So we tried to find important target transcription factors that are interacting with this mediator complex. So from the experiment, we found 68 proteins as candidates interacting with OSMED25, and we found one transcription factor, the MIB transcription factor, to be expressed at the at this region where early formation of lateral root is occurring. So we further confirm this tendency by real-time PCR or RT-PCR. So this is expressed at this zone wherein the lateral root type is decided. So we thought that the MIB gene is an important candidate target transcription factor interacting with the OSMed25. So in here, we further did an experiment to consider the interaction of the MED25 and the transcription factor MIB. And according to this experiment, we have here the control media, and we have here the control media without important amino acids for growth of yeast cells, and also it was supplemented with antibiotics. So this AD is serving as a negative control, and we can observe that yeast cells, we can observe that yeast growth can survive with MIB transcription factor, suggesting that MIB has really interacted with the MIB transcription factor. And so this MIB participates in the regulation of lateral root formation. And this is the yeast-2 hybrid experiment for the mutant, and we can see here that there was no interaction or there were no growth of yeast, so there was no interaction. So then the MIB interacts with OSMed25 in the regulation of the L-type lateral roots. So by utilizing these mutants, we have understood the mechanisms of L-type lateral root formation. So first is the mutation of the WEG1, which causes parental root bending that induce L-type lateral roots via auxin accumulation at the outer side of the bent region. On the other hand, the normal WEG1 gene regulates the straight parental root and produce S-type lateral root. So in the case of the PLR1 mutant, we have actually 3 mechanisms, although I only explained one mechanism. And one of the mechanisms is the more accumulation of starch in the stem induce less root sugar accumulation and produce S-type lateral root formation, whereas the mutation of the PLR1 induce less accumulation of starch in the stem and produce more root sugars and eventually induce the L-type lateral root formation. So another mechanism is less accumulation of root auxin in terms of the normal PLR1 gene, and then after that they produce S-type lateral root, whereas the mutation of this gene induce more accumulation of root auxin and produce L-type lateral root formation. And lastly is the involvement of the MIB transcription factor in the regulation of these L-type lateral roots. So I think this is a more interesting for you po. So for now, we are introgressing the WEG1 mutation gene to popular and high-yielding Philippine rice varieties, which are indicated in this breeding scheme. So we have already did this through funding from DOST. Then here we are showing clearly that the backgrounds don't possess this mutation gene, including the background of the golden rice, which is the PSBRC82. So the important advantage of this gene is that we have Now, the molecular marker for these improved lateral root rates that can be used for marker-assisted selection and therefore addresses the most challenging part of root breeding, which is the root phenotypic evaluation. So here we actually designed a specific marker for this mutation gene through a DCUPS marker. And because root breeding is a very tedious process, especially in the field, we established this screening protocol by making use of the root phenotype of WEG1. So we optimize this hydroponic system with combined modified rapid generation advanced setup. And in here, the WEG1 mutation gene is a recessive gene, so we make use of the 3:1 segregation setup. So we root phenotype the lines to without branching, moderately branching, and highly branching. And those highly branched root lines are the ones that we plant in this RGA setup. And we have a detailed experiment on this. And here is actually the actual photo of the RGA, wherein we have gradual or staggered planting selection of those WEG1 type lines from 3 different rice backgrounds. And from the RGA, we genotype our lines and selected the WEG1 type lines. Then we further select with the other desirable traits such as early heading, long grain, and the absence of on. So these are now our selection from F2, F3, and actually we are now in the F5 generation of our study. So a little bit summary of this presentation and the things that we are doing in our research. We hope that with the genetic improvement using these different mutation genes promoting root development, rice plant can increase its water and nutrient uptake under drought stress that consequently increase photosynthesis, thereby resulting to maintained yield and shoot dry matter production. So these are our co-authors and collaborators in this study. And one of the things that we are doing now is also doing the biotech application. So we are here at the DA Crop Biotech Center, and we are doing collaborative works with those researchers doing CRISPR-Cas to gene edit these genes to popular and high-yielding rice varieties. So for now, these are the things that we are doing in our research. And we are also expecting that our lines that we have developed will be evaluated in the field. And hopefully we can also have another funding for that for next year and the coming years, so that we can really test our lines in actual fields. So I think that this is a short summary, not really a short summary, but a review of the research that we are advancing and doing, especially in the case of root research, wherein we are trying to advance the underground revolution, which is a key to a second revolution.
Mr. Chair, may I ask a question, Mr. Chair?
Sure.
Yes, please, sir.
Yeah, well, Mr. Chair, as we know, I ask questions at the very end, but this is kind of exciting. So the two questions are simple. The first one is, what is your contact number, phone number, so people around the world can call you? Because I just want to let you know, she won this year the Outstanding Young Scientist Award, not from Rotary or anything like that, but from the National Academy of Science and Technology. That's very hard to get. So we want to get the phone number of the Outstanding Young Scientist Award for 2023. Would give us your number, even office.
Okay, sir. So my number po is 0961-057-8686 po.
Again, 0961
057-8686.
8686. So everybody around the world, this is the National Scientist Award for the youth, right? 0961-057-8686, right? Because, you know, a lot of stuff happens between farms. And the second last question is, first of all, no, we know that extension has really suffered in this country. When they got extension devolved to LGUs, most LGUs didn't do it. So we really suffered technology transfer. You have the best technology, etc. And we should spread it. I'd like to ask that question. How can we, listening to you right now, mostly here but around the world, how can we help you with your mission of bringing this kind of science, which is so needed, especially with climate change? How can we help you with your mission?
Yes po, sir. We don't want to do shortcut with our research. And right now po kasi the stage is to really do tests and evaluation in rice fields. And after that, if we have the concrete evidences that we can really use this for addressing the climate change resiliency or for drought and nutrient deprived soils, then I think you may help po in disseminating or in the use of these lines po.
Yes, we'd like to say this now. When you're done, you talk to us because we have very good contacts now.
Yes po.
We are in this council and we're in touch with other departments. And therefore, when you try, we'll help with your bureaucracy. One example we have is that there was a UPLB where you came from. Then some people waited 8 months now. So when we talked to them and they talked to the DA, they did it in 2 weeks. Similarly, this technology, when you're ready, when you spread it, it may not spread too far. But if you go together to the top people and they see the brilliance of young scientists, they'll do it faster. Yun lang po. So, Mr. Chair, thank you very much. Those are my 2 points. Please proceed.
Okay, thanks very much. So now we open our here for questions. Thanks very much for a very interesting topic, Nona.
Yeah.
Personally, I'm surprised that auxin accumulation can be concentrated. That was not something I was expecting to learn about today, because my normal experience with auxin is accumulation is above ground, not below ground.
Yeah, so yeah, I hope that I was able to give information because mostly most of the research, especially on rice, is on above grounds. So that's why I'm taking this opportunity to also share our research on below ground naman po. Yeah, and Sir Ernie, thank you so much for the comment. I really appreciate that we really need to also bring our research outputs, and actually not just outputs, the outcome of our research to our beneficiaries, especially our farmers who are most vulnerable in times of climate change. But yes, we are trying to make fast as much as we can. We are doing a lot of innovative works, especially on root research, which is a very difficult research right now, because we have to extract the roots from the soil, doing very tough work, something like that. So, but now we are trying to progress and innovate and improve as much as we can. So we don't have to really extract the roots from the soil now, because we can do the genetics and also molecular or the marker-assisted selection.
Hey, Mr. Chair, last comment. You know, we really are upset because our technology goes to Vietnam, Malaysia, Taiwan, and we don't use it here. And it may happen to you again. No? So we just want you to know that we're businessmen, and we have to make sure that technology reaches our people. And the fact is that 70,000 farmers disappeared in a sense, right? 7,000 extension workers, because they were gone. But there's new MEP ABCD is very involved in that, the transfer of technology. As a matter of fact, that's the reason for being transfer of technology. Thank you, Mr. Chair.
Thank you, Professor.
So yeah, we move on to our open forum. So as always, we'd like to limit open forums to just one question. Maybe one follow-up if in case the follow-up is very relevant to the first question, especially for clarifications. What you can do is you can look at the bottom of your screen if you're in computer. You should see a button that says reactions. Click that, and then there's an option that says raise hand. So click that, and you will see that your hand is raised like Oscar's is right now with the little yellow hand on top. If you're on your phone, you can look at the three dots or the three horizontal lines, depending on whether you're on iOS or on Android. Tap that. That should show you reactions. Tap that, and then you'll see raise hand. Tap that. Otherwise, you can put your question in the chat box, or failing that, I'll ask Bernie or Vivian to please help me to catch my attention if someone's trying to ask a question on screen and I can't see that. Okay, Doc Nona. So thank you very much for that, and I'll call Oscar, please. Oscar, your question. You're on mute. Oscar, you're mute.
Okay, sorry. I'd like to ask if your technology involves any DNA splicing, you know, something like CRISPR. I've learned from the internet abroad that they're using this CRISPR method of DNA splicing, even on plants and fruits. So I'd like to ask you that. Thank you.
Yes, sir, for the mutation genes, for its application, especially in case of biotech, we will employ the CRISPR-Cas, but this will actually be done by my colleague here at the ACRAP Biotech Center. Yes, sir. We will use the CRISPR-Cas in order to put this gene to our popular rice varieties.
Yeah, okay, follow-up question, no? Okay. Well, this DNA splicing has caused a lot of controversy. There are a lot of people who are saying that we are, you know, manipulating Mother Nature, and maybe there are many negative side effects and things like that. So I suppose for you, you're more on the science part. Are you prepared to counter, you know, objections of that sort? Yeah, thank you.
Yes, sir. So I would like to just give emphasis that using the CRISPR-Cas technology is different from doing genetic transformation, because in the CRISPR-Cas technology, you can only target the base sequence wherein you can do change only base naman po. Like for example, in our mutation gene, we only have a single nucleotide substitution or single base change. So I think it will not be a very controversial method to introgress genes to other plants using the CRISPR-Cas technology, unlike doing the classical genetic transformation before, because you have to put a large chunk of the genetic information from other organisms to a new or to your target or recipient organism. Yun po, sir. And actually, I have mentioned about mutation. So it actually occur in nature. So in the case of CRISPR-Cas, if you are only manipulating a certain base or few number of bases, I think it's the same with the mutation that is occurring in nature. Yun po, sir. But that's for my perspective po.
Yeah, okay, thank you.
We have another question from Melchor Virata.
Yeah.
Hi, sir. Good
morning, Nonawin. Nonawin, your technology is more on under the ground, right?
Yes po, sir. But We try to create lines po, sir.
Okay. Have you checked or I checked with BARD the total land area of the lakes, about 1,771 square miles. That's equivalent to 240,000 hectares of water, fresh water. Have we tried working on it, on fresh water? I saw a technology. I have no idea whether from China or from Vietnam. Styrofoam, and then they put rice plant. So the roots, 24 hours have water. Have you tried that? Have you researched on that? That's 240,000 hectares of water, 24 hours all over the Philippines.
Sir, can I follow up a question?
Okay, thank you.
Is
That's
it
all.
like a hydroponics kind of cultivating the
Yeah,
rice
I think so. They tried it on rice.
Yes po, sir. We haven't yet considered really a big cultivation of rice in hydroponic system pa po. Although we are doing it for our small experiments and also for advancing our rice lines po. But I think that is also a very interesting aspect of cultivating rice, especially now that we are trying to as much as possible expand the cultivation of rice in different methods and ecosystems po. Sir, about is it the lake po ba, sir?
Yeah. I think any where the fresh water are located.
Yes po, sir. If we can also do expansion of our rice cultivation kung where rice is where water is available.
In Vietnam and China.
Yes po, sir.
Okay.
We hope we can also consider those kind of cultivation
That's all.
methods po.
Okay.
Mr. Chair, just want to say this. So we want to give you a request. We want you to bring this up to your management, what Melchor said, because Melchor is, in other words, this is dyadic, we help you, you help us. Melchor is brilliant, and please bring it up to your management. We'll follow the up within 3 months. You bring it up, and then if necessary, you call Melchor, right? In other words, this is a give and take, right? You give, we take, but sometimes we give and we ask you to take, and that's one suggestion. Melchor is saying, look at it, no? So can you do that? Bring it up to your management.
Okay, sir. Let's try to discuss po. We will try to discuss with our core researchers.
And the secret of management is follow up. So we'll follow up with you in 2 months to see what happens. Okay, Melchor, ganda sinabi mo. Salamat po.
Okay. Sorry. All right. Next we have a question from Ike.
Yes, Dr. Agustin. Is drought-resistant variety suitable for upland growing also? And is the government identifying upland areas so there will be no more need for irrigation?
So, yes po, sir. Upland area is also considered as a drought stress-prone environment, and we also need to consider breeding upland rice varieties or rice varieties suitable for that upland ecosystem po. So just to put our research, we are also considering on improving rice varieties that will be suitable for upland rice ecosystem by enhancing the root system formation, especially deep rooting and then lateral root branching ability. Kasi the ones that we have before only have the deep rooting, but they don't yet possess the branching ability. Then in here at field rice, upland rice is also considered in our research, although most of the researchers now are focused on the big rice production in our country, which are the irrigated and also the rain-fed lowland po.
Okay. Are there other questions
Julius,
from
if
the
none, I'll
audience?
ask.
Sure, Bernie, go ahead.
No, now my question is, in the researches that you are doing, are there inputs that you get from farmers in terms of their needs or aspirations?
Inputs from farmers. I'm currently expanding my research works, and recently we tried to go to farming communities. For example, in Ifugao, they have their heirloom rice, and one of the things that we are doing is learn from how they cultivate culturally their rice, and how can we integrate um, and also how can we integrate their cultural management and the modern way of farming that we have in rice management po. One interesting aspect that I've learned from them is on uh, on improving different traits of the rice. Although when we were there, it was not related on root rates, but more on reducing the crop duration, and also increasing the stem number of rice so that their productivity will be increased. So it depends on the rice ecosystem that we are visiting. In rain-fed ecosystems naman po, for example, even around here in Nueva Ecija, wherein we pass by different rice fields, there are really cases or increasing cases that rice fields are now droughted at some point of the crop duration. So I think that is really one of the inspirations. That's why I'm pursuing my research to address the drought vulnerability of our rice fields, even not only on rain-fed lowland, but even the irrigated rice areas. And actually, even our irrigated rice areas practicing the AWD or the alternate wetting and drying strategy to conserve water consumption of rice. But these plants are also exposed to drought stress at some point. For example, mild stress, because you have to allow the rice fields to of course alternate wetting and then drying. You allow the rice fields to become droughted at some point, but this actually have effects on rice growth already.
Okay. Well, I can this race, but I'll give way to other people who'd like to ask questions first. I also have a question, but I'll save it. If are there any questions? No, okay, Ike, go ahead.
Yeah, I'm just actually working on my common sense. If you already have drought-resistant varieties and you already have upland rice varieties, why doesn't the government make a policy to plant all areas that do not have irrigation? For example, in Mount Pulag, there are hundreds and hundreds of hectares there, and nothing grows there except grass. In Mindanao, there are so many mountainous areas that do not have irrigation, but they are rain-fed, and in some places, like in Bukidnon, it's already in the clouds, and it's fed by mildew and mist. So why not make it government policy, very simple, not scientific, but whenever you have areas that do not have irrigation, just plant your variety, doctor. Plant drought-resistant and upland rice. Thank you.
Yes, Pastor, thank you for the suggestion and comment. I think it's really one aspect that we have to consider, especially those areas na sayang naman po if we don't plant crops po, no? If rice or drought-resistant varieties can be suitable to that area, why not plant? So I think we can also consider that in terms of, for example, policy. If we have in Ifugao, when we went there last time, their rice varieties are Tinawan rice, their heirloom rice, and there are areas that they that are not already planted with any crops. So what the mayor did was to make resolution and instruction to their office to really plant rice to these areas which can be suitable for rice planting, but they were abandoned by different inhabitants already. So I think we can do it po, sir, because it is working in Ifugao already.
All right. I'll just ask my question and then we can wrap up for this session. Yeah, the research is honestly very, very interesting. My question is that because I'm trying to think about it from the perspective of the business application. First, it's research. There's a few steps before we can get it commercial. But from that perspective, like to sort of help me understand like the mechanism that allows it, to say that it's drought resistant. So I'm guessing that the auxin accumulation is not actually the major factor. It just happens to be the sort of one of the correlations to the changes you're making. It's actually the accumulation of sugars because you did mention in one of the slides that it seems to be that auxin accumulation is what causes the waviness of the roots, but also part of that, sorry, correct me if I'm wrong,
Yes,
it also causes a larger amount of, or sorry, not causes, it's related to a larger accumulation of root sugars. And that's what allows it to resist drought more. Is that correct?
Yes, po, sir. So let me just say the auxin accumulation in the root is important to induce the L-type lateral root, which is our target trait in order to allow the branching ability of the roots. So in the case of the sugar or the carbohydrate mechanism, which I mentioned, what is the relationship of the sugar and auxin? Right now po, we are still doing the research. Which is first, the sugar or the auxin? We don't know the answer po, sir. But the auxin is important to induce the L-type lateral root formation, and at the same time, the accumulation of sugars, especially glucose and sucrose in the roots, are also important to induce the L-type lateral root formation. Yes, po, sir. So the first
So,
we don't have answer.
okay, that's fine. I was trying to sort of understand how that mechanism affects the drought. Is that something that can be seen in other plants as well, or is that mostly just in rice?
That mechanism po, sir. So if I may compare it to our model plant, which is the Arabidopsis. Arabidopsis is only producing one type of lateral root, and that is L type. So in the case of rice, it's very unique because it has two types of lateral roots, the S type and the L type. So to say that is it happening to other plants, in Arabidopsis there is report on the auxin accumulation to produce L-type lateral roots. So I think it's similar to rice. But in the case of sugars, There are also reports on Arabidopsis wherein if you put sugars to the roots of the Arabidopsis, they produce highly or much more number of lateral roots. But Arabidopsis has only one type of lateral roots, while rice has two types of lateral roots. Yes.
Thank you so much for the clarification because I was trying to sort of understand. In our side, for example, research that we've been looking at through the Philippine Seed Industry Association is in gene expression and silencing. What we found is that certain plants in the pursuit of drought-resistant or drought-tolerant crops, what we found is that gene expressions that help the plants, in our case, is vegetables. So, for example, tomato. So not rice. It's different than that. But I wanted to share this because it's related to like the question and like does this have something to do with the expression of drought resistance or tolerance? In crops that have expressions, gene expressions that help it resist drought further, what we found is that unfortunately it comes at the expense of certain other factors that or other traits that most farmers would find desirable. So for example, the yield decreases, stunted, things like that. Which makes sense because you know you're planting it under harsh conditions.
Yes.
So obviously when those gene expressions are there, you know, the plant is trying to protect itself. So research on from seed companies, for example, has been mostly on is it possible for us to activate the gene expression, like we silence the gene expression for drought tolerance or drought resistance when there's no need for it? And then is there a way for us to say activate that expression? I don't know if I'm using the right scientific terms. I'm not, but the idea is that we only use it when it's necessary, like if we know that there are and things like that. So it's very interesting. So so thank you very much for that. It's very interesting that it's really more trying to figure out a way to improve the root system's ability to
we actually have similar thinking on like when are we going to activate this gene and when not to activate this gene. So it's maybe it's easy to just think and say that, but when you do actual manipulation on plants, it's not easy po, especially when there are different signaling mechanisms that are affected. Because sometimes one gene can doesn't only affect one signaling mechanism, it can affect different signaling mechanisms. For example, the one which I presented, the OSMED25, it's a protein complex. So it attaches to other proteins and they are regulating different mechanisms for not only the signaling mechanisms, but for example, the defense mechanisms, the drought tolerance mechanisms, and other traits that the plant has. So it's very important that we need to understand the basic, for example, for example, the signaling mechanism, which is more important, the sugar or the auxin signaling mechanism, so that we know what to prioritize for the plant to respond to drought, something like that. Yes, but I think if we can have that technology, we can try.
I'll link you to the people who are working on it. You might be able to exchange notes on. I'll try to see if I can reach out to them and see. Sorry, we went a little bit over time. Maybe Ernie, I can ask you to say a few words before we end our formal session. Or
just to tell you, I like the youth taking over the country. You're better than the old people, so please proceed. And we want to tell you, we want to do something here. So we want action from this, and I want to tell Melchor to just follow up. We'll help Melchor. Thank you very much.
Thank you so much, sir.
Anna, maybe you'd like to say a few words just to close out.
Yeah, so I would really, I really appreciate this invitation to share our research. It's really one of my passion. share what are the advancements that we are doing in our research, and we hope that they are insightful to other researchers, so that we can somehow do some collaborations later, because everything is interrelated naman po. So I'm really very thankful, and I appreciate this invitation. And hopefully we can think more on how to collaborate so that we can better utilize the knowledge or the information that we are generating, and also the research outputs that we are producing with our research. So thank you so much.
Okay. And so to end our session for today, may I first present our certificate of appreciation to Doc Nona. Yeah, so the Agribusiness and Countryside Development Foundation, in partnership with the Management Association of the Philippines, together with all of our members, our partner institutions, special partner institutions, senior special partner institutions, corporate members, and individual members, all present this certificate of appreciation to Dr. Nonawin Lucob-Agustin for her invaluable support in the advocacy and promotion of agribusiness and countryside development, and for commendable and unselfish services rendered to this foundation as our resource speaker, given on our 27th weekly online meeting of 2023, this 25th day of July, in Makati City, Philippines, with an audience from all over the world. Signed myself as chairman, and Ernie Ordonez as our president. Now, may I please invite everyone to turn on your cameras, go into gallery view, and ask everyone to turn on your cameras, show us your wonderful faces, bright and early in the morning. Please, cameras on, everyone. Melchor, Elsa? Oh no, Elsa, I think had to go ahead. Robert, Yupanto, Yvette, I, Tingling, yeah, everyone. There's somebody here named ABCDMA227. If you could very kindly like turn on your camera, name yourself. Okay, yeah, guys. Medyo shy yata yung mga ibang kasama natin. Yeah, Robert's here, Tingling's here, great. Clarice, yan. Oh, Clarice is on her way somewhere. Sorry, Clarice. All right, Vivian, could you count us in, please?
All right po, we only have one page po. At the count of 3, 1, 2, 3. Thank you po.
Okay. Well, for those of you who do have to go ahead, okay, well, anyway, and then for those of you who are interested in next week's session, can you just flash next week's session on screen, just so everyone can see that? So next week's session will be about a smoke-free Philippines by Dave Gomez of PMTFC as the communications director. So hopefully this has been a topic, I think, related. This is a topic that's related to one of our previous topics regarding the state of the tobacco industry in the Philippines, and it might be good for us to get some insights on the consumer end of things, especially considering that there is a strong anti-smoking movement, anti-smoking legislation that's has been implemented over the last couple of years. So that might help us gain some insights into the consumer side of things. Okay, so with that, thanks very much, Doc Nona, for all of your time. We invite other people to stay to listen to our past performance, but I will have to ask you to leave by the time we get to some more sensitive parts.
Okay, thank you so much, Mr. Thank you, everyone.
And Dr. Nona, if there's a need for continuing interaction, let us stay in touch.
Yes po, ma'am. Please feel free to be in touch with us po.
Thank you.
Thank you so much. Thank you so much for the invitation.
We will be in touch for sure. I'm gonna call you up for sure.
Okay po, sir. We will be waiting.
Thanks so much, Doc.